So welcome to the Transfer to Excellence Research Symposium. We're really happy that you're all here. We are so thrilled, please ignore the presentiment view, that we've been able to run a program this summer, given everything that's happening globally. We're so grateful to our interns and our PIs that have hosted our wonderful remote interns. And together we've navigated what does it mean to do a remote hands-on research internship. And it's been working. It's been going really, really well. And the interns have had, in my understanding, a great time and learned a lot. Some logistics, we have muted everyone because this event has been widely shared and we want to prevent Zoom bombing. However, we will have time after each presentation to ask questions and to discuss the research. So please use the chat. Sam will be moderating for questions and asking them. And you can also ask to be unmuted and we'll unmute you so you can ask your questions. And so we'll be starting this morning with Mimi, who will be talking about her work in Professor Boker's lab. And then you can see our planned agenda here. Going on to Cameron and then V. Wyatt, Musael, Rosario. And we'll be wrapping up with Samantha and Austin. So without further ado, I'm going to open things up to Mimi Winchell, who has been working with Professor Boker and Dr. Sucheta Mandal this summer, who is going to speak about her research. Mimi, you should be able to unmute yourself, I think.
Yes.
And let me share your slides. Is that showing the right slides?
Yeah, that works.
Oh.
Okay, thank you. Good morning, everyone. My name is Mimi Winchell, and I'm going to share a little bit about the research I've done this summer in Professor Boker's lab with my mentor, Dr. Sucheta Mandal. The topic of this research is current-induced switching in an anti-ferromagnetic system. And before I go into the specifics about our research, I'm going to introduce the background of anti-ferromagnets and magnetic switching. So next slide, please. So since technology is rapidly advancing, there are a few goals scientists have for the 21st century of data storage technology. So the first is to achieve faster magnetic switching. And they're also trying to miniaturize data storage devices and to have more compactly stored data. And another aim is to reduce energy consumption. So current semiconductor devices require high amounts of energy to work. So researchers are looking for suitable alternatives to reduce energy consumption. And this summer, we've been working with anti-ferromagnets as a possible alternative material. Next slide, please. So that brings us to the question of why anti-ferromagnets? So Louis Niel, the scientist who actually discovered anti-ferromagnets, described them as interesting but useless. But in more recent years, researchers have been focusing more on anti-ferromagnets as a replacement for traditional magnetic materials. Anti-ferromagnets have many properties that can facilitate more robust and non-volatile designs for data storage. So first, anti-ferromagnets don't have stray fields, so they're robust against external magnetic fields. And they also possess high precessional frequencies in the terahertz regime, which allows for ultrafast dynamics and ultrafast switching. And similar to ferromagnets, we can use spin orbit effect-based magnetotransport to manipulate the Niel vector, which is the vector parallel to the directions of the spins in the antiferromagnet. And I put a little diagram to the side of the antiferromagnet picture, which shows the Niel vector. And finally, there's a wide variety of antiferromagnets, including metals, semi-metals, oxides, and insulators, and that allows for more flexibility in materials. So the main drawback to antiferromagnets is that they're difficult to read and study because of their lack of net magnetic moment and their insensitivity to external magnetic fields. Next slide, please. So the images here show some of the most important advances using antiferromagnetic materials, including direct and indirect techniques for reading, which is in the pink diagram, and writing, which is the green diagram. It's by electrical means. And so antiferromagnets are usually used as passive elements in these structures, such as in the spin valve system and the magnetic tunnel junction. More recently, direct switching of antiferromagnets is being used for designing magnetic memory elements, such as the one in the image to the right, which shows a multi-level memory element. Next slide, please. Okay, so in this project, we created two nanostructures. First, in the top left image, we layered nickel oxide, which is an antiferromagnet, on top of platinum, a non-magnetic heavy metal. And in the second structure to the right, we added a layer of permalloy, which is a ferromagnet, on top of the previous structure. In both cases, we injected a current pulse through the heavy metal layer. And this charge current was converted into a spin current, and it exerted a spin torque on the interface between the heavy metal and antiferromagnetic material, so between the gray and orange portions of the images. And this torque acting on the interface actually compels the antiferromagnet's Neal vector to rotate. So in the images at the bottom of the slide, we have examples of 90-degree and 180-degree switching of the Neal vector. So after the current pulse is injected into the system, the arrows inside the nickel oxide, which is the antiferromagnet, they rotate, and that's known as switching. Next slide, please. So we created a nanostructure of 50 nanometers by 50 nanometers per layer. And by using this nanodot, this small size, and by using a low current, it provides more energy efficiency. Okay, next slide, please. Okay, so I'm going to introduce the micromagnetic simulation that we used. Next slide, please. Thanks. Okay, so now I'll give an overview of how we achieved the previously mentioned aim of this project. So first, we created a numerical model of the system using a micromagnetic simulation software called UMF, which stands for Object-Oriented Micromagnetic Framework. And at the bottom of the slide, there's an image of how the simulation looks in the software. So the red and blue layers of the antiferromagnet have anti-parallel intra-layer coupling, meaning that they're like going in opposite directions. And in the one with the ferromagnet, which is FM, we placed that on top and layered it parallel to the top layer of the antiferromagnet. So the second step in our process was to observe the static properties by varying the applied magnetic field, which is also known as the Zeeman field. We did this so that we could observe the extent to which the magnetic moments would align in the direction of the applied field, and to see the susceptibility of antiferromagnets to this field. So this portion of the project is mainly to verify the effects of antiferromagnets and ferromagnetic coupling within the system. And the last step in the process was to explore the dynamics of the system after the current pulse was injected into the non-magnetic layer in the absence of magnetic field. So we took away the magnetic field that we used before, and now this is just with the current. And this is the main application of antiferromagnets as possible materials for data storage, because by reading the direction of the Neal vector, the data could be understood by the device. Next slide, please. Okay, so now I'll go through the static magnetic properties that we observed. Next slide, please. So these are images of the magnetic hysteresis loops of the two systems. So on the x-axis, we have the Zeeman field in milliteslas, and on the y-axis, we have the magnetization, and that doesn't really have units. So before I begin, I'll just explain the term saturation. Saturation is when all the domains in a magnet are pointing in the direction of the applied field. So in the heavy metal antiferromagnet system, which is on the left, within the range of applying negative 10 Tesla to 10 Tesla, which is a pretty strong field, the sample never saturates, meaning that the magnetic domains stay mainly pointing in opposite directions. And this shows the strong anti-parallel coupling within the system and shows that it's a reliable antiferromagnet to use. And this also displays the antiferromagnet's insensitivity to the magnetic field, which I discussed in the introduction as a crucial component for non-volatile design of storage technology. So in the heavy metal antiferromagnet hysteresis to the right, the sample reaches saturation pretty quickly after we apply the magnetic field. And in the zoomed in figure in the corner, there's approximately a 30 millitesla shift of the origin to the left. So usually the hysteresis would, the center would be right above 0, but now it's shifted to negative 30 millitesla. And this shift depicts the exchange bias that's present because of the coupling between the antiferromagnet and ferromagnet. Next slide, please. Okay, so now I'll go through the antiferromagnetic switching that we observed. Next slide, please. Okay, so after we finished testing the static properties of the system, we took away the applied magnetic field and injected current through the nanostructure in the X and Y directions separately. So I put a little diagram of what it looks like in the top corner. Current pulses were injected with different pulse widths. So for example, the red pulse lasted for 100 picoseconds, whereas the green pulse lasted for 1 nanosecond. So in the time verse magnetization graph on the right, the high frequency precession in the terahertz range is shown in the close-up picture in the corner. Since the Neal vector spins are compensated, no clear magnetization switching is visible on this graph, but the oscillations show that there is excitation during the current pulse. Next slide, please. So these graphs over here depict the dynamics of the heavy metal antiferromagnet system after injecting the current pulse. In the red and blue data, which both have low currents, the ferromagnet doesn't show magnetization switching in the time verse magnetization graph. The lines dip down, but then they return back to the original positions mainly. And the green and yellow data, on the other hand, correspond to higher currents, which you can see on the time verse current density graph. In the time verse magnetization graph, the green and yellow colors switch their magnetization directions. Next slide, please. Okay, so over here, we'll see the effects of pulse amplitude versus pulse width. So in the green graph, we have a switching map due to current applied in the Y direction through the heavy metal antiferromagnet system. And in the blue graph right next to that, we have switching through the same system, but with current applied in the X direction. And in the yellow map, we have switching due to current through the Y direction in the heavy metal antiferromagnet system. And in the orange, it's through the same system, but in the X direction. So we varied the current density from 10 to the 8th joules per meter squared to 10 to the 14th joules per meter squared. And we also tried a range of pulse widths from 1 picosecond to 5 nanoseconds. So 1 picosecond is 1 times 10 to the negative 12th seconds, and 5 nanoseconds is 5 times 10 to the negative 9th seconds. And here are a few observations that we've found. So first, it takes a smaller amount of current to switch the antiferromagnet than to switch the coupled antiferromagnet and ferromagnet. So starting as low as 10 to the 10th joules per meter squared, the antiferromagnet was able to switch, but we actually observed no switching through the antiferromagnet-ferromagnet system at that current density. And we also see a trade-off between pulse width and current, which plays a large role in the probability of switching. So with a smaller amount of current, both the antiferromagnet and antiferromagnet-ferromagnet systems require larger pulse widths to switch, But on the flip side, when an extremely high current is applied, such as at 10 to the 14th joules per meter squared, a shorter pulse width is needed to produce switching. So one of the main takeaways from this data is that it's easier to switch the systems with broader pulses at a smaller expense of current, which is actually corroborated by a literature in this field that we've read up on. Next slide, please. Oh, okay, so we ran into technical difficulties, but this was actually supposed to be a video visualizing what 90-degree switching looks like in our actual system, but sadly, I won't be able to play that today. And so do the next two slides. Yeah, okay. So to summarize, antiferromagnets, we saw that they exhibit strong coupling, and antiferromagnets coupled with the ferromagnet displayed an exchange bias. And those 2 points were seen through the hysteresis loops. Both 90-degree and 180-degree switching was observed from that switching map that we just saw. And spin current driven antiferromagnet switching consumes less energy than currently used in CMOS data storage devices, which is what we use today mostly. And in the future, which is really exciting, using antiferromagnets for data storage will allow for more non-volatile memory and more dense storage. So there's an image to the right, which shows a closed pack array of antiferromagnets, which it's just showing that we can more densely store data using these antiferromagnets. Next slide, please. Okay, so here I give some references of the articles we've read, the papers, and also of the OOMF system that we used. Next slide, please. Okay, so I'd like to thank Professor Boker, who was my host PI this summer. I really enjoyed working in your lab or remotely in your lab. And I'd like to thank Dr. Sucheta Mandal, who was the most amazing mentor. She was really supportive and amazing throughout the whole process. And I'd like to thank the Transfer to Excellence research internship at the University of California, which is this program. And Nicole and Sam and everyone, you guys were amazing. So thank you so much. And funding for this project was provided by the Center for Energy Efficient Electronic Science, which is a National Science Foundation Science and Technology Center. Okay, next slide, please. So thank you so much for watching. And are there any questions?
Just a couple of questions. And this is more of out of curiosity. Your experiments were essentially based on using nickel 2 oxide with platinum. And then my curiosity is, did you ever try nickel 2 sulfide? And if you did, what was the near vector effect on it?
So we actually only used nickel oxide in this experiment because we had read some articles and they had the exact like properties that we should plug into the simulation software. So and we were also going to, if we had time, we were going to try IRMN, but we haven't tried the one that you were speaking about, sadly.
Okay, thank you. And my next question is, what are the limitations of the OOMF system?
Sorry, could you repeat the question?
The limitations of the software program that you use, the OOMF system.
Yeah, so it's an LLG simulation, so it solves the Landau-Lifshitz-Gilbert equation, which I didn't put over there, but yeah, so that's how it solves the problems. So it uses a mathematical model to simulate, and then it displays it, so it gives back all of the data, and so you just have to be like really careful about which numbers you're putting into it. And yeah, so that's how we used it. Does that answer the question?
I don't want to put you on the spot. My curiosity is, you know, with all these computer programs, you put in garbage and you end up with garbage. So I'm just wondering what the factors or specific limitations associated with what you use.
Yeah, so actually, the reason we did this numerical model is so that hopefully later on, Sucheta, which is my mentor, or someone else in the lab could actually verify these results using like real live samples. So for now, we are just creating the model, and it can mainly back up what they could find later on. And it's an atomistic simulator for the antiferromagnet, meaning that we got like really close. So we modeled the system based off of how an actual antiferromagnet would be in real life. Just using this software instead of like actually using a sample.
Thank you.
Thank you.
I think Wyatt has a question.
Yeah, I just had, could you maybe explain like what the difference between the 90 and 180 degree switching is, if there's like an advantage or disadvantage between the two?
Yeah, so actually the main thing that we'd want to happen is 90-degree switching, because since the Neal vector points in both directions, so whether it's like at 0 degrees or 180 degrees, the device couldn't really like know which direction that's happening in. So we're trying to get 90-degree switching. Obviously, we didn't get it at all of the conditions, but so instead of like having something that's like flat, we could have some the Neal vector, which is pointing up and down instead of to the left and to the right. So that's mainly like that would be the best results. So 90 degrees is the aim for antiferromagnets when using them to create other devices.
Thank you.
Any other questions for Mimi?
Yeah, I had a quick question.
Oh, you go first, Mason.
You want to go first? Okay. My question was there a reason specifically why the anti-ferromagnet was between the heavy metal and the ferromagnet?
Yeah. So like I mentioned in the introduction, so antiferromagnets don't have like stray fields, and so it's kind of hard to read them. So the ferromagnet, which is placed on top, it actually, so when it switches, you can read that. So it's hard to actually read the antiferromagnet by itself, but when the ferromagnet is on top and it switches too, then we could read that data.
Okay, thank you.
I have probably a basic question.
I was wondering why an antiferromagnet is called an antiferromagnet and its difference between itself and a ferromagnet.
Yeah, so a ferromagnet, so when it's in a magnetic field, all the sublattices will align in the same direction. So for antiferromagnets, it just like means anti. So like one will be pointing up and one will be pointing down, or one will be pointing to the side and the other one will be pointing to the other side. So, and they're just compensated spins. So instead of like having, all in the same direction, they're all like in compensated direction. So there's no net magnetization.
Thank you.
Thanks.
Awesome. Well done, Mimi, especially with internet problems. We appreciate you.
Thank you.
We had Cameron Zara Baker scheduled to go next, and we're having big technical problems. So my apologies to everyone from Dr. Kate's lab. We are trying to get her back online ASAP, but we're gonna move ahead to VTran in the meantime, just to keep things moving, and we'll get to Zara ASAP. So V, you should be able to share your screen. And go ahead and take it away.
Hi everyone, my name is Dee, and I'm from Orange Coast College. And today I'm gonna talk about my research, which is unsupervised deep learning for lensless imaging. This research is conducted in Professor Laura Wansler's lab, mentored by Grace Puel and Christina Monacover as part of the TTE REU program 2020. So traditional camera use lens to refract light rays onto the sensor. So however, lenses are usually the component that takes up the most weight and volume of the system. So with lensless imaging, we replace the lens with a diffuser, which is made from a thin, transparent piece of lightweight and low-cost material like plastic or masking tape. So hence the imaging system can be very compact and cheap. A unique properties of lensless imaging is that it can map a point source in the field view to many pixels on the sensor. So as we can see here, if we place a flashlight in front of the camera, we'll get a caustic pattern on the sensor, and this is called a point spread function. And this point spread function is very important for the reconstruction. So lensless imaging has many applications. Using lensless architecture, we can build a very compact and imaging system for microscopy. So here's a prototype of a 3D fluorescent microscopy that can capture the 3D fluorescent volume. The lensless imaging integration helps to reduce the volume and the weight of the system. Another application for lensless imaging is in compressive sensing. So as a point in the real world maps to many points on the sensor, we can pack more information onto a sensor reading. As it is demonstrated here from a still image that we have here, we can reconstruct a high frame rate video with lensless imaging system. If we use lensless camera to take an image of the real world, the immediate result that we get here is meaningless, and we'll call this as the sensor measurement. And we need a reconstruction algorithm to get back the meaningful scene image. So using the point spread function, we can make the forward model that takes in the characteristic of the diffuser and establishes a relationship between the sensor measurement and the reconstructed image. So in practice, we'll get a point spread function and the sensor measurement from the camera, and we'll use this forward model to solve for the scene image. So currently there are two approaches used for reconstructing images. The first method uses a model-based approach. This approach formulates the inverse problem as an optimization problem and solve it using gradient descent with FISTA or ADMM. So as we can see on the graph at the bottom, the lowest point on the graph is the scene image that we hope to get to. And this point here can theoretically converge to the desired point after many iterations. However, with this method, the equation has a hand-tuned prior, and this value is different for each images, and there hasn't been any systematic way to find it other than trial and errors. And this is the current limit of this method. The second approach takes an unsupervised deep learning approach to reconstruct images. So this method incorporates the physics model of lensless imaging with a deep network to automate the hand-tuned prior using a large data set of ground truth images and sensor measurements. And as ground truth images are hard to get with the lensless imaging, we seek to have a better reconstruction method for lensless imaging system. So in this research, we explore the territory of unsupervised deep learning for lensless imaging as it addresses the limitations of other approaches. So why unsupervised deep learning? This approach is appealing as it replaces the hand-tuned prior with a deep network that learns by itself, and it doesn't require any ground truth images or retrained network. We can solely use the point spread function and the sensor measurement to reconstruct the scene image. So here's a diagram that illustrates each step in this method. Firstly, a random input is generated and fed into a deep network. The network output, which is the reconstructed image, will go into the forward model, initialized by the point spread function to create a simulated measurement. We'll then calculate the distance between the simulated measurement and the actual measurement using the last function to assess the network performance. This value is then used to update the network to produce a better reconstructed images. After many iterations, the system will arrive at the meaningful scene image. About the first step, the deep network is a series of operations that transform the input into something else. So in this model, we ask the network to update its weight parameter to represent the reconstructed image. We will test two different network architecture called Deep Image Prior and Deep Decoder to compare their performance. Both networks are untrained and have randomly initialized weight parameters. Deep Image Prior has more layer and more complex structure, and hence having more weight parameters to learn. We call this an over-parameterized network, which means that it has more parameters to learn than the number of pixels on the image. The second architecture is called the Deep Decoder, and this network is simpler, have less layer, and less weight parameters to learn, and we call this an under-parameterized network, which means that it has less weight parameters than the number of pixels on the image. For the loss function, we want to calculate the distance between the simulated measurement and the actual measurement. We can directly calculate the mean square error of these two images and use it as the mean square error loss. However, with the mean square error, the network tends to favor the low spatial frequency of the image and give a result that are blurry and lose the fine details. So here we implement another loss function to compensate for that. First, we transform the image into the frequency domain in which the low spatial frequencies will gather at the center of the image. Then we will generate a mask that has a low value at the center and high value everywhere else. And we're going to apply this mask onto the frequency transformation to get this image. Well, and we will calculate the mean square error of these two transformations. For the experiment, we use images from two sources. The first example images from the data set that were taken in previous experiment taken in the lab and that required a precise setup. A natural scene image is displayed on the monitor screen and was captured by a lens camera and a lensless camera. For this source, we have a lens and a lensless version of the same image. We will use the lens image to test the performance of our approach. The second data set is taken at a more casual setup I had at home with books, masking tape, and my phone flashlight. I took images of a real object using a lensless camera. For this one, we only have the lensless image, which is the sensor measurement. Here's the result that we get using this technique. As the network is randomly initialized, the first iteration is meaningless. But after more iteration, the network will be able to learn more about the measurement statistic and performs better. At the bottom is the graph that shows the difference between the simulated measurement and an actual measurement across iterations. In general, the smaller the difference, the better the result. After a couple thousands of iterations, we'll be able to see the general structure of the image, and with more iterations, we'll be able to retrieve the fine details. Here we compare the unsupervised deep learning method, deep image prior, and deep decoder with a model traditional based method called FISTA. So generally, unsupervised deep learning is able to reconstruct the scene image, although their performance seems varies with different networks. Here we have a FISTA reconstruction after 300 iterations, the deep image prior after 10,000 iterations, and deep decoder after 10,000 iterations. Deep decoder reconstruction seems to be a little bit noisier than the deep image prior, and their performance seems to be on par with FISTA. Besides comparing the result visually, we also use the mean square error and the output score between the lens image and the reconstructed image from 3 methods to compare their performance. With these metrics, the smaller the value, the better the result. We can see here that FISTA has the highest mean square error and outputs among the 3. So both deep decoder and deep image prior performs better than FISTA. Among the other 2, deep image prior performs better than deep decoder with a smaller mean square error and output score. But in terms of reconstruction time, FISTA remains the fastest method. Here are the results for the experiment images with real objects that I took at home. Here's the image of the Rubik's Cube, sub-animals, and a set of markers. So visually, the deep decoder seems to perform better than the deep image prior. Both FISTA and deep decoder are able to retrieve the fine details of the image, while the deep decoder, while the deep image fire is quite blurry. To compare the between deep image fire and deep decoder, I plotted the mean square error and upper score over 25,000 iterations. The orange line represents deep image fire and the blue lines represent deep decoder. As the orange line always stays below the blue line, for the butterfly image, the deep image fire performs better than the deep decoder. Here's the progression of frequency loss and mean square error loss of deep image fire. As these random iterations, frequency loss image has more details on the butterfly wings, and more resembles the original scene image. We can observe the similar behavior from the deep decoder, with the frequency loss seem to help the network to converge faster to the desired scene image than the mean square error loss. But generally, both loss are able to converge to the similar result. However, using experiment images I took at home, the frequency loss gives out a very different reconstruction, with a frequency loss result seems to be more interpretable and more resemble the original scene image. As you can see here, the mean square error loss and frequency loss are converging to a two very different results. So in conclusion, we tested the performance of unsupervised deep learning approach for lensless imaging. The result is almost as good as the traditional method. Over-parameterized network and under-parameterized network behaves differently, with the deep image fire performs better with the sample image, and the deep decoder performs better with experiment image. And by modifying the loss function to account for the high spatial frequency, the network converges faster and more into a more interpretable result. So here are some of the failed run I got before I arrived at a good reconstruction. And for some images, it just doesn't work at all. So the network still requires some hand tuning for the hyperparameters. And in the future, we hope to automate those values, like the learning rate, the number of parameters for each layer, or the number of iterations. I want to thank the computer imaging lab, especially my principal investigator, Professor Laura Waller, my mentors, Grace Puo and Christina Monakova, for allowing me to conduct this research. I would also want to thank Nicole McIntyre, E3S staffs, for providing support throughout the program. This research is funded by the Hopper Dean Foundation.
Awesome, well done. Thank you, V. Any questions in the chat? We got some applause. Let's give you one more minute. So just to remind everyone to avoid Zoom bombing, we are avoiding automatically unmuting everyone. So please put your questions in the chat or ask to be unmuted. Austin would like to know, can you talk more about the loss function you applied to your images, V?
Yes, so for the loss function, the general approach to calculate the distance is that you take the pixel-wise value of two images, and then take the subtraction of them and square them and take the mean of those values. But with that approach, the result was kind of blurry and don't have the fine details. So here we account for the high frequency more. So we transform the image to the Fourier domain so that we can gather the high frequency, so that we can gather the low frequency at the center of the image, and we'll multiply it with a mask. That mask has a low value at the center and high value everywhere else. So the high frequency of the image will be way more than the low spatial frequencies.
Thank you. And Professor Boker has a question.
Yeah, well, I have two questions. Early on in your talk, by the way, V, very nice work. I love this project. I was a little puzzled by one thing that you showed near the beginning. Maybe I wonder if I understood it correctly. It looked like you showed that this type of approach could allow you to encode a video in a single still image. Did I understand that correctly?
Yes. So this is something that a regular lens could not do. So as one of the first image I saw with a point spread function, a point in the real world matches to a lot of points on the sensor. So if we remove some of the points in the measurement, we can still see kind of the pattern on the sensor reading, and with just a little points that we got over there, we can get back the image. So another way to address this to answer is that we have more points than we need to reconstruct the image. So basically, we can pack more, we can encode more light information onto one single sensor exposure.
Do you have any idea how many images you could encode into one? How long a video could this be? You know, 100 images or 1,000 images or?
The project earlier I saw is the project of another of Nick Antipas, another graduate student in the lab, and like I didn't I'm not the person who execute the research, but we are exploring the application of deep image wire and deep decoder with the compressive sensing to see if we remove some of the measurement if we remove some of the information on the sensor measurement, can we also reconstruct the scene image just like when we use the traditional model-based method?
Okay, fair enough. Back on your project, the you know, at the end you were talking about different frequency domains and different algorithms, how they might work. I wonder if you have thought about the actual optical element that you're using to substitute for the lens. So it's just some kind of random ground glass, right?
Yes.
And would there be, have you thought about, would it help to try to design that element in order to try to, you know, optimize some of these issues that you're talking about with respect to different frequency components?
We haven't got to that part yet. Currently, we are using like a very simple off-the-shelf materials to do that. But with the diffuser, the lab are already like designed a kind of a diffuser to use for the microscopy that I mentioned at the beginning. So with a design diffuser, we will be able to probably improve the possibility, also the capabilities of the lensless imaging system.
Very good, thank you.
Awesome, well done. I want to remind you all that these interns have been doing all of their research remotely. Many of them have never been to Berkeley. They've never met their mentors or their PIs in person. They've done all of this in 8 weeks online, which is very impressive to me. Up next, we have Wyatt Jabeth, who also interned in Professor Boker's lab. And Wyatt, you should be able to share your screen now.
Okay, perfect. That. Okay. And then, let me see. Is that the full screen? Let me try that one more time.
Yeah, that was your full presenter mode.
Okay. Okay, there we go. Let me just move this down a bit. All right. Okay, good morning, everyone. My name is Wyatt Jabeth. I am a summer research intern with the Transfer to Excellence program at UC Berkeley, and I'm a rising sophomore at Santa Barbara City College. And over the last 9 weeks, I have worked closely with my mentor, Dr. Zafer Mutlu, with supervision from my PI, Professor Jeffrey Boker, to better understand the Raman spectra of atomically precise graphene nanoribbons. And today I'm excited to share our work with you all. Okay, I'm going to quickly go over the outline of this talk. First, I will give an overview of graphene nanoribbons and discuss the core motivation for this work. And next, I'm going to describe the methods used in the study. And finally, I will discuss our results and findings. And so I'm going to start by giving a background to graphene nanoribbons. So some of you may be wondering, what are graphene nanoribbons? Graphene nanoribbons, or GNRs, are quasi-one-dimensional carbon-based semiconductors that possess a tunable band gap contingent on ribbon width and edge geometry. Simply put, they are ultra-narrow strips of their parent material graphene. And graphene is a two-dimensional material comprised of a single-layer hexagonal carbon lattice. And so this figure on the left shows 3 different GNR edge designs: armchair, chiral, and zigzag. And as the electronic properties of these ribbons are directly tied to their edge structure and width, these structures are incredibly important. And also in this figure, here I'm going to turn on my laser pointer. Just a second. You can also kind of see this hexagonal structure that I mentioned. And so at each of these points would be a carbon atom. Okay, and this middle figure describes the inverse relationship between armchair GNR width and their band gap. And in this study, we primarily focus on armchair type GNRs because they are more stable in ambient conditions. And so this figure shows that as you increase the ribbon width, you lower their band gap and vice versa. And this final figure on the right shows a simple model of a transistor in which the GNR acts as the channel between 2 metallic electrodes. And because GNRs are incredibly small and have a tunable band gap, they are a promising channel material for future transistors. Okay, but why is all this important? Well, over the last several decades, silicon, which is the standard channel material for modern transistors, has been pushed closer and closer towards its hard physical limit. And consequently, a new material is needed to ensure the continuation of Moore's law in the looming post-silicon electronics era. So why have GNRs not made it to industrial applications? Well, there are many challenges currently hindering GNR technology, but one of the most urgent is their large-scale characterization. Because GNRs are generally less than 1 nanometer in width and about 20 nanometers in length, commonly used characterization techniques such as atomic force microscopy, AFM, and scanning electron microscopy, SEM, cannot be used to study GNRs. And so today, there are only 2 standard methods for characterizing GNRs: scanning tunneling microscopy, or STM, and Raman spectroscopy. And so although STM is an effective method to obtain atomic scale information about GNRs, its scope is localized and is limited to samples grown on metallic substrates. And on the other hand, Raman spectroscopy is a fast and non-invasive analytical approach that provides complementary information about GNRs on both metallic and insulating substrates at a macroscopic scale. However, most contemporary studies that use Raman spectroscopy only do so to identify GNR type. And so the primary focus of this work is to better understand the Raman spectra of GNRs in order to improve both their large-scale characterization and implementation in future transistors. Okay, and now I'm going to describe the methods we use to grow and transfer GNRs. And additionally, I will provide a brief explanation of Raman spectroscopy and reiterate its importance to GNR characterization. So there are many ways to fabricate GNRs, but the bottom-up approach is preferable because it provides ultimate control over ribbon design and electronic properties via engineering of specific molecular precursors. And in this study, we use the molecular precursor dibromobianthracene, or DBBA, to synthesize 7 atom-wide armchair GNRs, or 7A GNRs for short. And this figure on the left is a simplified illustration of our ultra-high vacuum system in which we grow our ribbons. And so to grow 7A GNRs, we use a heating and annealing cycle to evaporate DBBA molecules onto this gold thin film, which rests on this supportive mica substrate. And so as the DBBA molecules are evaporated and deposited on this thin film, they simultaneously polymerize. And so once polymerization is complete, a final heating cycle induces cyclodehydrogenation, and atomically precise 7A GNRs are formed. And so this figure on the bottom right is an STM image from a paper previously published by members of the Boker group that shows 7A GNRs synthesized using this method. And as you can see in this inset figure, their edge structure is smooth and defect-free. And so to summarize, this bottom-up approach is essential because it enables GNR bandgap engineering through molecular precursor design, and it produces uniform GNRs ideal for device implementation. And so after our 7A GNRs are successfully synthesized on our gold mica sample, we use a wet transfer method to move them to a device-ready silicon substrate. So first, we place our sample in a hydrochloric acid aqueous solution in order to delaminate this mica substrate. And so once the substrate delaminates and sinks to the bottom of our solution, we use a pair of tweezers and a steady hand to lower our target substrate onto the GNR gold sample, top face down. And then once adhered, we take the sample and we place it into a potassium iodide-based gold etchant. And that'll kind of etch away the gold thin film, leaving just our GNRs on top of our target substrate. And once this sample is cleaned, we also inspect it for creases induced during this liftoff process. And we use generally a droplet of isopropanol to flatten these wrinkles and further adhere the GNRs to the silicon substrate. And once transfer is complete, electron beam lithography is used to deposit palladium and form electrode channel contacts to complete the device. And because our GNRs now rest on an insulating substrate, we can no longer use STM to characterize them. And this is where Raman spectroscopy can be incredibly effective. And so here I'm going to introduce Raman spectroscopy and explain the ways in which we use it to study GNRs. And so this figure on the left shows a simplified schematic of our Raman spectrometer. Essentially, we shine a green laser or monochromatic light at our sample, and the light that is reflected back generally has the same or less energy as our laser. And so this change in energy occurs when molecules of our sample, after being excited by the laser, do not return to their ground state, but settle at some vibrational state. And so the difference in energy between the incident laser and the light that is scattered from our sample is plotted as a graph like the one shown here on the right. And so this is the Raman spectrum of 7A GNRs grown on gold 1-1 that we recorded using a 532 nanometer laser. And so each peak is representative of a specific characteristic of our sample material. So for example, the G peak is caused by carbon-to-carbon bonds, while the D peak over here is a result of the ring-like lattice structure of GNRs and the abundance of GNR edges detected in each Raman sample. And so additionally, these inset figures describe the actual atomic displacement of their corresponding vibrational modes. And so in this study, we primarily focus on the D and G peaks, as well as the ratio of their respective intensities. And so now I'm going to discuss our experiments and results. And so over the last several weeks, we have investigated substrate and doping effects on 7A GNRs to better understand the ways in which the wet transfer method alters GNR electronic structure. And additionally, we have sought to identify relationships between these effects and shifts observed in the Raman spectrum of our GNRs. Okay, so first, we use the standard bottom-up approach I described earlier to synthesize atomically precise 7A GNRs on both gold 1-1 and copper 1-1. And so after analyzing the Raman spectra of both samples and comparing our results with additional Raman data of 7A GNRs grown on other metal substrates, including gold 7-8 and silver 1-1, we identified a shift trend for both the G mode frequency and the D to G intensity ratio, which is shown here in this graph on the left. And so, yes, and so in the case of graphene, shifts in the G peak are an indication of lattice strain, which can cause changes to the graphene's electronic structure. And as I mentioned earlier, GNRs are simply ultra-narrow strips of graphene. So their electronic properties are very similar, their lattice structure is very similar. And additionally, this figure on the right illustrates the effect of gold 1-1 on the band gap of isolated GNRs. And so for metals with different band structures, the electronic interaction between GNRs and the metal substrate will vary. And as a result, the change in GNR band gap will vary as well. So as you can see, these are theoretical calculations. So for GNRs, isolated GNRs, the calculated band gap is about 3.7 electron volts. And then once these GNRs are on a gold 1,1 substrate, the band gap decreases to about 2.3 or 2.7 electron volts. And so with further research, we believe the G-shift trend shown here may be correlated to changes in GNR electronic structure via substrate effects, including both electronic interaction and strain caused by lattice mismatch. Regardless, based on these results, shifts in the G mode frequency and the D to G intensity ratio clearly indicate change in GNR electronic structure. And so next, we doped our 7A GNRs grown on gold 1,1 with hydrochloric acid, or HCl, and diluted potassium iodide, Ki2. And so these are the two primary chemicals used during the wet transfer process. And the intention of this experiment was to analyze the isolated effect of these dopants on GNRs. And so this figure on the left shows the normalized G mode of doped and undoped GNRs. And here we observe the same G-shift trend discussed on the previous slide. And so this result further confirms that a shift in the G peak can be attributed to change in GNR electronic structure, and in this case, via doping. And this middle figure describes the change in both the line width of the D peak and the D to G intensity ratio. And so for GNRs and other similar carbon-based materials, the D to G intensity ratio is a measure of sample defects. And additionally, the D peak is a result of, like I said, the GNR's ring-like lattice, but also their edges and imperfections. And so therefore, increases in both the D peak line width and the D to G intensity ratio indicate an overall increase in defects. And so based on these results, it is clear that HCl is cleaning and purifying the GNRs during wet transfer, while Ki2 is in some way damaging the GNR structure and altering their electronic properties. And so this final figure on the right provides just additional evidence that we are actually doping with these chemicals. And so this plot describes the gate potential and drain current relationship, or on-off ratio, for a GNR-based transistor after being doped with HCl. And so for graphene and carbon nanotubes, a shift in the minimum drain current indicates doping. And as you can see here, for just our standard GNR transistor, we have this minimum off spot close to the 0 gate voltage. And then after being doped with HCl, it's shifted up to about 0.5. And so that's a clear indication of doping for materials such as graphene and carbon nanotubes. And we believe that this is showing the same effect here. Okay, and then finally, we analyzed the Raman spectra of 7A GNRs before and after wet transfer to an insulating silicon substrate. And so these two plots show our results from all three experiments. And on the left, the G-shift trend is again observed after transfer to silicon. And on the right, we show the changes in D line width and the D to G intensity ratio. And in both plots, the shifts observed after doping with Ki2 and after wet transfer to silicon are very similar, and they're circled with these blue circles on the graphs. And so based on this result, we believe that the Ki2-based gold etchant used during the wet transfer process is one of the primary causes of transfer-induced defects. And so to summarize our findings, we have shown that Raman spectroscopy has applications beyond GNR fingerprinting, and that with further research, Raman may be used to probe substrate and doping effects on atomically precise GNRs. And additionally, we have shown that potassium iodide, or Ki2, is harmful to GNRs, and an alternative etchant or transfer process should be explored. And in future studies, we hope to use STM imaging to confirm the Raman trends noted in this work. And so now I would like to give a special thank you to my mentor, Dr. Zafer Mutlu. Without his guidance, patience, and support, this project would not have been possible. So thank you, Zafer. This is also an image of him giving me a lecture on electron behavior, and he's using these two toilet paper puppets to demonstrate that. I thought it was pretty awesome. And then I would also like to thank the following people, Nicole and Sam, along with the entire TTE staff, for making this remote experience a reality. My PI, Professor Jeffrey Boker, for trusting in my ability to be a contributing member of his group, and the other Boker Lab Group members for including me in your discussions and meetings these last 9 weeks. Professor Michael Bartol for taking the time to engage my quantum curiosities, and my fellow interns for your friendship and support. I hope we can all meet at some point in the future. I would also like to acknowledge the Center for Energy Efficient Electronic Science and the National Science Foundation for funding this research, as well as the Molecular Foundry for providing the equipment necessary for this project. And so with that, I would like to thank you all for listening. If there is a question I cannot answer or do not get to, please feel free to contact me via email. Thank you.
Very well done. Thank you, Wyatt. Any questions from the group?
Yeah, I just have a quick one.
Yeah.
Out of curiosity, so all the research that you have presented, were they done online using some kind of simulation software?
No, so yeah, so it's all been remote. So the bulk of the work that I've done is analyzing the Raman spectra of these ribbons grown on all sorts of different substrates and doped in different ways. The actual growth and transfer of the ribbons is performed by my mentor in lab up in Berkeley. Yeah, and so I mention it as a way to kind of describe the process and what we're doing to these ribbons. Yeah, but there's no software used. It's all using real samples. The only theoretical calculation was the figure that I showed showing the GNR band, like the isolated GNR band gap, and that was performed by a different group. Yeah, I hope that answers your question.
And just my last one, in terms of reproducibility, how did you find the literature on it?
The stability?
Reproducibility, how reproducible?
For growing the ribbons or for analyzing them or?
For growing the ribbons.
For growing the ribbons, yeah, so there have been several studies. So the bottom-up approach that I mentioned during the talk, I believe it was first shown in around 2010. I can check that. But yeah, it's a reproducible approach. It's been used many times. And so our group uses it because it provides this sort of uniform ribbon that is ideal for implementing into different devices.
Any final questions?
Yeah, of course.
Yes, I have a question.
Yeah.
So you in the speaking, you said we sometimes experience the wrinkles on the films.
Yes.
And I'm saying, oh my God, that means he got into the lab and he actually experienced it. But actually, this was communicated to you as something that sometimes happens by Mutlu. Is that right?
Yeah, that's correct. Yeah, ideally I would have been in the lab. No, but my mentor was incredibly supportive. He actually showed video of himself in the lab performing the transfer using the tweezers and picking up the substrate and the ribbons.
But our labs were closed, so maybe he snuck in.
Yeah. No, so yeah, so that video was from like I think it was before pre-lockdown. So
pre-lockdown.
Yeah.
Okay, I had another question. Yeah, I had another question. You had as one of your dopants HCl. Now, if it was HC and lowercase l, then I know what that is. That's hydrogen chloride. But I didn't sound like this was hydrogen chloride. So what was HCl with the uppercase L?
Right, it was hydrochloric acid. That might have been a typo on my part.
Okay.
Yeah, I apologize for that confusion.
Okay.
Yeah,
All
it
right.
was just
Thank you.
It's the same, hydrochloric acid that we used during the wet transfer. Yeah, so we could kind of separate the different effects that we were observing.
Final question for you, Wyatt, from the chat. Lakshika would like to know why SEM was not used to study it again. SEM.
SEM?
Yes.
Okay, that is a great question. I don't have an exact answer for that. I know that in our group, we were primarily using STM to study the ribbons after growth on metal substrates, and that once we transferred to an insulating, we were only using Raman. But I would be happy to engage in an email correspondence. I could get some more information on that. That's a really good question.
Awesome, thank you. Very great answer. With that, we are gonna move along to Cameron Baker. Cameron or Zara has experienced technical difficulties this morning that were not her fault and stayed very gracious and cool under pressure. So Big props to Cameron, and thank you to the folks from Professor Kate's lab for sticking around. I think you have an 11 o'clock lab meeting, so we really appreciate you, especially Angelica, for being here for this presentation. Cameron should be able to share their screen. I believe that Zara will be presenting from her phone, so thank you in advance for your grace and patience. Looks great, Cameron.
Okay, sorry. So good morning, everyone. My name's Karen Baker, or you may know me as Zara. I'm a student at Fullerton College, and this summer I was working as a Transfer to Excellence intern working on a research project in Professor Jamie Kate's lab with my mentor, Angelica Gonzalez-Sanchez. And the project we were working on was investigating sequence features of EIF3 and EIF4A target mRNAs. So to begin with, we're going to talk about essential dogma of molecular biology. So this is a two-part process in synthesizing proteins from DNA. The first part of this process is going from DNA to RNA and is called transcription. The second part is translation, which is going from mRNA to proteins, and this is what the Kate lab studies. Translation regulation is critical for maintaining cellular homeostasis, and its misregulation leads to diseases such as cancer. So here in this image, we see translation, and it consists of 3 steps: initiation, elongation, and termination. Translation initiation is a complex process. It's the most energetically demanding cellular process. It's highly regulated, and it requires several eukaryotic initiation factors known as EIFs for short. So here's an up-close image of the first steps of translation initiation. And here we can see one of the most pivotal EIFs, EIF3, which acts as a scaffold or as a bridge in pre-initiation complexes. This is one of the EIFs I will be focusing on for this project. So you may wonder, why is EIF3 so important to understand? Misregulation of EIF3 has been linked to many diseases, such as 19 different types of cancers, 4 types of neurodegenerative diseases, and 3 types of infections. Here's an image including these diseases, and some of them include like breast cancer, Parkinson's disease, and hepatitis C. So data from the Lee and collaborators' 2015 paper here is shown. And in this publication, it was shown that beyond the role of a scaffold, a novel role for EIF3 is that it regulates translation of a subset of mRNAs. Moreover, EIF3 can act as a repressor or an activator in mRNAs. In the case of June, which is an mRNA that is specifically studied by my mentor, it acts as an activator. As well, the data shows different processes and functions that EIF3 target mRNAs that are involved in, such as cell cycle, DNA repair, and RNA binding. So data from this paper also shows that EIF3 predominantly interacts in the 5' UTR. So here's a structure of an mRNA. It consists of the cap, the 5' untranslated region known as 5' UTR, the coding sequence known as CDS, and the 3' untranslated region in a poly A tail. So this is where EIF3 is interacting in the mRNA. From the same paper, we see the secondary structure for June's 5' UTR, and in this region where EIF3 interacts in the mRNA, it's called the Parclip site, and at the boxed region, this is a site, and you can see that it is in this secondary structure of a stem loop. So with all of this information, the Kate Lab continues to investigate EIF3 in different contexts, one of which is the context of the multi-factor complex known as MFC for short. So in the previously reported MFC, it was shown to be composed of EIF1, 2, 3, 5, and methynyl tRNA. However, the Kate Lab has found a novel human MFC that was shown to be composed of EIF2, 3, 4A, 4G, and methynyl tRNA. So given that EIF3 regulates June's translation, and it was seen in the human MFC of June that EIF3 and EIF4G, it poses that do EIF4A and EIF4G play the role in June's translation regulation? So here are the first steps that we saw before of translation initiation. And here we're looking at EIF4A, another critical EIF that we will be focusing on in this project. EIF4A unwinds mRNA in preparation for recruitment to initiation complexes. So in 2016, Izwaki and collaborators published a study about rocoglamide A, known as ROCK-A, a compound that clamps EIF4A onto specific mRNA sequences. Their study showed that June was highly sensitive to ROCK-A, meaning that it was an EIF4A target. As well, they showed that GAAG and GAAA are target sequences of EIF4A. So given that June is an mRNA of both EIF3 and EIF4A, the question my project focuses on is, can EIF3 and EIF4A regulate translation of other mRNAs? So from the two papers that were previously discussed, a cross-reference was put together of the EIF3 targets and the EIF4A targets, and it was found that there was 116 mRNAs that were targeted by both. And of this 116, there were 37 that were highly sensitive to ROCK-A. So the objectives that my project focuses on is to investigate the sequence features of a subset of mRNAs that are targeted by both EIF3 and EIF4A, and to find these common features and hypothesize a mechanism for translation regulation of these mRNAs. So to begin with, we started with a functional analysis using David Bioinformatics Analysis database and on the 37 mRNAs, and we found that there was 18 common functions of the mRNAs. Some of these functions were ones that were previously reported in EIF3 target mRNAs, such as cell cycle, DNA repair, and RNA binding. So we chose to focus on a subset of 21 mRNAs that were found to have the common functions of RNA binding and nuclear processes for further analysis. So while looking at the sequence features of these mRNAs, we saw that there were different isoforms, which are slightly different versions of mRNAs that have different lengths. For the rest of this analysis, we chose to stick with the ones that were most validated, which are the most prominent in the cell. Using these isoforms, the length of the annotated 5' UTR was recorded, and as you can see in this graph, that the lengths of the mRNAs were found to be around 150 to 350 nucleotides. So the Parclip site data from the Lee and collaborators paper was analyzed, and we saw that for most mRNAs, there was one Parclip site where EIF3 interacts. However, with HN, RNPAB, U2AF2, and TMEM33, we found that this has two sites. This means that EIF3 can interact significantly in more than one site in some mRNAs. Then using the integrative genomics viewer, we did a sequence. The sequence of the Parclip for each mRNA was found and mapped on genomic DNA using SnapGene. The length of the Parclip site was recorded, and we found that most fell to be around 30 nucleotides long. So by mapping the Parclip sequence in the genomic DNA using SnapGene, the location of the Parclip was found, and for each mRNA, most were found to be in the annotated 5' UTR, meaning that their sequence was reported as part of the 5' UTR in databases. However, some Parclip sites were found in other regions, and some of these regions were not reported in databases, but since we observed them in the Parclip raw data, we can say that they are unannotated. For example, some of the Parclip sites were found in unannotated 5' UTR, annotated CDS, unannotated CDS, and a few fell into areas where they were partially annotated 5' UTR and partially unannotated 5' UTR. So due to the discrepancies in the Parclip location, the 5' UTR length had to be corrected for some of the mRNAs using the raw Parclip data. So in the blue, you can see this was the annotated 5' UTR, and then the orange is after we revised it, and it greatly increased the length for the mRNAs. So then the EIF4A target sequences GAAA and GAAG that were reported in the Izwaki and collaborators paper were counted per region, so on the 5' UTR and on the CDS and per mRNA. And it was seen that for most mRNAs, there seemed to be a greater amount on the CDS opposed to the 5' UTR. So one possible factor that there was more EIF4A target sequences in the CDS opposed to the 5' UTR could be due to the fact that the CDS is much longer than the 5' UTR in most RNAs. So then the amount of EIF4A target sequences were quantified in the regions going 200 nucleotides 5' of the Parclip site and 200 nucleotides 3' of the Parclip site. And this graph illustrates that there are no EIF4A targets on the Parclip site, which is where EIF3 binds, and that the majority of EIF4A targets were found after the Parclip site. So just to remind you, it was previously reported by Lee and collaborators' paper that June's Parclip site where EIF3 binds was found in a stem loop. And so because of this, we decided to look into the secondary structure predictions of some of the mRNAs we focused on in this project to see if there are similarities in the Parclip site secondary structure predictions as seen with June. So here are some of the most common secondary structures found in RNA and that we took into consideration for this analysis. So there's a single-stranded region, the stem, which is just this part, the bulge loop, the interior loop, the hairpin loop, also known as a stem loop, which is including all of this, so the stem and the loop, a junction, also known as multi-loop, and a pseudoknot, as well as there is an exterior loop, which is not shown here. So then using the University of Vienna RNA fold web server and the Matthews Group RNA structure prediction web server, the secondary structures predictions were found for both the Parclip site and the EIF4A target sequences within plus or minus 200 nucleotides of the Parclip. So here it is shown that for the secondary structure predictions for the Parclip, they were found mostly in stems and stem loops. And then for the EIF4A target, they were found in a greater variety of structures, with the interior loop being the most predominant, followed closely behind by the stem and stem loops. So here is an example of a secondary structure prediction using the Matthews Group web server of BRD2's 5' UTR. And here in the red box is the Parclip site, and it is predominantly shown in a stem loop. And then here is one of the EIF4A target sequences, GAAA, which is shown in a stem. So some conclusions that we can take from this analysis is that the mRNAs that were both targets of EIF3 and EIF4 had many common functions, including some that were previously seen in EIF3 target mRNAs, such as nuclear processes and RNA binding. Most of the Parclip sites were found to be around 30 nucleotides long, giving a rough estimate of the sequence lengths that EIF3 binds to. As well, we saw that most of the Parclip sites were found in stems and stem loops, suggesting that EIF3 binds to highly structured areas. Some other conclusions that we had were that the CDS contains more EIF4A target sequences than the 5' UTR, which is potentially due to the substantial difference in lengths. While the Parclip sites in most mRNAs were found in the 5' UTR, supporting the Lee and collaborators 2015 data, EIF3 was also seen to interact in other regions of the mRNAs, such as the CDS. As well, none of the EIF4A target sequences were found on the Parclip site, and the majority were found upstream of the Parclip site. And lastly, we saw that the EIF4A target sequences were found in a variety of secondary structures, which can be potentially explained by the fact that EIF4 scans mRNAs rather than binds to them like EIF3. So as a whole, this study broadens the understanding of the mechanisms of translation regulation mediated by EIF3 and EIF4A, which serves as a blueprint for targeting disease. And some people I would like to acknowledge is my mentor, Angelica, and my PI, Professor Jamie Kate, as well as the E3S and the TTE REU staff, especially Nicole and Sam, especially just because it was at a very uncertain times and all of us were very anxious to see whether we were going to be able to participate in this program or not because of COVID and lockdowns, and they made it happen. And then as well as my fellow interns for just all the support. Daily check-ins with you guys were great and it always made my day. And then this work was funded by the National Science Foundation. Sorry, it won't let me stop screen sharing. Okay.
Very well done. Any questions for Zara? Feel free to put them in the chat or raise your hand.
While those questions load, I had one really quick. I thought the annotation mention was really interesting. Just as like a non-scientist, I think it's neat that from the sound of it, you can kind of edit the nucleotide length of these genes almost. And I'm wondering sort of if you could go into that a little bit more and like how that was done or what the results of that were.
So yeah, we went into like the Parclip raw data from the paper that was seen, and using the integrative genomics browser or viewer, we were able to like input the data that was shown, and then you were able to like see these peaks of where the interactions were happening. It just didn't show in the databases as annotated 5' UTR, but it was there. So we labeled it as unannotated.
Okay, sure, thank you.
Any other questions? Okay. My understanding is that Zara did such a great job this summer. The scope of the project was expanded to more mRNAs than previously planned, so that's awesome. Ted, who has previously interned in Professor Doudna's lab, is wondering how your research could prevent cancer-related diseases.
So the lab that I was working in, they do more of the investigation and then publish the data so that others can then use that information to be able to do those things like figure out like medications and different things that will happen. But the lab is not a bench to bed lab, so we do not interact directly with that. We just publish the data so others can use it and benefit.
Awesome. One more question from the chat. Samantha says, you mentioned ELF4A and ELF4G in the beginning. What has been done with ELF4G?
So EIF4G, so we saw both EIF4A and 4G in the novel human multi-factor complex. It's just, it hasn't been investigated yet, and so that's for following work to be done.
Awesome. Thank you so much, Zara, for your hard work. And also, I just want to thank Sam Mountain for your hard work getting Zara back online and troubleshooting our technical problems.
Yeah, thank you.
With that, we are going to move along to Musael Gebremariam, who is going to be presenting on his work in Professor Ming Wu's lab. Awesome. Great. Take it away, Moose.
Sorry, I forgot to unmute myself. Hi, everyone. My name is Musael Gebremariam, and for my TTE project, I worked in Professor Wu's lab, supervised by my mentor, Xiaosheng Zheng. And for the past 9 weeks, my research project has been about the design and optimization of LiDAR beam scanners. So first, I'd like to introduce what LiDAR is. So for LiDAR is a light ranging and detection system in which a laser is illuminated onto the target, and that reflection of the laser is shot back into the receiver. And with this method, we're able to determine the distances of the objects in our surroundings by using this equation here. And to our right is a GIF of a LiDAR application on an autonomous vehicle where the LiDAR is mounted onto the car and it's able to detect the other objects in this environment. So for the types of beam scanners that we analyzed in this project, we analyzed mechanical and solid state beam scanners. For the mechanical side, we specifically analyzed a galvo mirror. And the way this works is a laser is shot into the first mirror, and since it's able to be deflected onto the second one, and since each mirror has a motor in which allows it to rotate, we are able to steer the beam in 2 dimensions. And for this scanner right here, this is just an example of a commercial scanner used in industry that's from the company called Velodyne. And for our solid state beam scanners, we specifically analyzed the focal plane array, but an optical phase array is another scanner that falls into this category. And the key emphasis I want to make for this project is that we wanted to first analyze how we can improve the current commercial galvo mirror products. And for our solid state beam scanners specifically, we want to know how we can implement them into the next generation of for our LiDAR application. So for our focus for first part, since like I said, how my project was split into both the mechanical and the solid state beam scanners. For first, we analyzed the galvo mirror, and the question that we wanted to be answered was, with a given light beam diameter, what is the smallest size for each mirror that we can achieve? And for the second part, with a given galvo mirror commercial product, what is the biggest beam diameter that we can be used? And following that, how much mirror area would be wasted? So for our approach for our galvo mirror is demonstrated through these 2 diagrams. And what we have here is the mirror that is in this right here is denoted as the angle of rotation for mirror 1, and we just said that would be alpha, and same for mirror 2, and that is called beta. And this is just the light beam cylinder, which is incident on the axis of rotation for the mirror. So once it's deflected from the mirror, it's no longer able to move. So here is just the height is just the distance between the first mirror and the second mirror in our setup. So for our mirror shapes, we see that when a light beam is incident onto the mirror while it's at an angle, it creates a elliptical cross section. And since we know that this is the behavior of the cross section, we were able later to determine that the angle that controls the mirror also controls the size of the ellipse. So that is the shape for the first mirror. And a big difference between the first mirror shape and the second one is that the second mirror is going to have to be able to capture all of the possible ellipses that follow between the reflection angle of the first mirror. So the angle, the reflected angle range that we have here is just theta between 90 degrees and 150. So we approximated, the shape of the second mirror to be a rectangle, just so it would be big enough to capture all of the possible ellipses. But we weren't able to get this into our, during the 9 weeks of our research. But one proposal that we made was that, if we're able to use a computer program such as MATLAB, we'd be able to map out the infinite amount of ellipses that fall in between this angle range. So for our mirror size analysis, we measured the mirror dimensions with respect to the angle range. And as you see here, this blue line, the minor axis length stays constant because for that elliptical cross section, the minor axis is equivalent to the diameter of the cylinder. And these are the other dimensions of the ellipses that change as well. And so for when we analyzed our commercial Galvo product, we first wanted to determine the relationship between the angle range and beam diameter of both mirrors in our setup. And since the angle ranges are not the same for mirror 1 and 2, we deduced that there could have been a mechanical limitation of the Galvo mirror. And for our for the second plot to the right, we see that what I measured was the percentage of area used with respect to the beam diameter. And we see that with a 10 millimeter beam diameter for mirror 2, it achieves a 60% mirror area efficiency. So for our findings of the Galvo mirror, we're able to see that the most efficient mirror shape for mirror 1 is an ellipse. And for mirror 2, we use the rectangular approximation, but it can be improved with the assistance of a computer program. And for our commercial beam scanner, we see that since both mirrors did not have the equivalent angle range, there could have been a problem in the mechanical setup of the Galvo mirror, such as the electrical circuitry or the clashing of both mirrors. And for the second part of the project, we analyzed the focal plane switch array for our solid state beam scanner. And for our focus for this part, we wanted to know which alignment errors do we need to take account of in order to achieve precise optical alignment, and how will we measure them in our experiment. And with the knowledge of these alignment errors, how can we address them in our optomechanical package that we design, and how can it achieve good mechanical reliability and have a compact size. So for our focal plane switch array, it consists of a 2D focal plane array and a lens. And the way this works is light is able to enter through the input port through an optical fiber, and a MEM switch is able to control the direction at which the light is able to travel. So once the light is able to travel through, each row, there is a grating at the end of each of these couplers. And once light enters each grating, light is able to be emitted into free space. And since we have that lens located at its focal length, then that light will convert into a parallel light beam. So here's just a demonstration of how each grating represents a different output beam angle. So this shows that this is a discrete function and not a continuous one. So for our approach for our focal plane switch array, we used a geometrical optics model. And with this approach, we're able to calculate the output beam angle by using a inverse tangent relationship. And in order to measure the beam behavior, when we do assess these alignment errors, we used a infrared camera and a second lens. And with this image right here, it just shows how how the camera is able to read that light beam and how it's displayed on that image. So for our first data result, it was for our alignment error, it was the out of focus scenario. And what this means is the chip is offset a distance from the focal plane, so it's no longer on the focal plane anymore. So when the chip is offset a distance below the focal plane, we denoted that as a positive change. And when the focal plane is, or when the chip is offset a distance above the focal plane, we denoted that as a negative change. So that's why for their corresponding graphs, for the converging angle, it's positive, and for the diverging angle, it's negative. For our next alignment error that we analyzed, it was a lateral offset. And what this means is that the chip is, the axis of the chip is no longer aligned with the axis of the lens. And what this does is it creates an asymmetrical angle range on both sides of the chip. And to the graph to our right, it just shows how amount of distance that we offset the axis of the chip, and how does that affect the beam angle. And for our last alignment error that we analyzed was a tilted angle. And what this means is that the chip is no longer on, it's tilted with an angle that we said was alpha. And what this causes is that since the ends of the chip are no longer on the focal plane, it the out of focus scenario is also part of this alignment error as well. So that means that since the left side of the chip is under the focal plane, that'll cause a converging beam, and since the right side of the chip is above the focal plane, that'll cause a diverging beam. So like I mentioned earlier, the way we measured this alignment error was by using an infrared camera and a second lens. And this diagram below here is just one of the possible scenarios that could happen in which the camera or the light beam converges before it hits the camera. So after the light beam converges, it begins to diverge. So when the image that the camera will read will be a spot size that will be bigger than what we saw previously in the other slide. So the way we measured, the way we measured using this camera was by seeing the two following alignment errors, which was the out of focus situation and the tilted angle situation. And since we were able to, since we have knowledge of what the alignment error does to our beam, we implemented those into our package design. And these are the designs that we did in Fusion 360. And here is just a C-mount lens, and we chose this lens because it was the most fitting for our chip size. And this ABS plastic holder is screwed into this printed circuit board. And what this does is it creates, mechanical stability between the camera and the chip. So there isn't any alignment error involved. So for our findings of the focal plane switch array, we're able to see that, with those different scenarios of alignment error, it also changes the behavior of the parallel light beam. And for our package design, we were able to achieve, those optical requirements. And, with that mechanical stability of the plastic holder and being screwed into the printed circuit board, we were able to eliminate any alignment error. So that was my project. I'd like to say thanks to my mentor, Zhao Sheng, and my principal investigator, Professor Ming Wu, and for their invaluable guidance. And I'd also like to say thanks to the TT staff, Nicole McIntyre and Sam Mountain, for their support along the way. And I'd also like to say thanks to NSF for funding this project. So thank you for listening, and I hope you learned something from my research.
Nicole, you're muted. Yeah.
It was bound to happen sooner or later. Thank you and well done, Musael. I'd like to encourage you all to ask questions. In the meantime, I'm going to remind you that all the presenters you're seeing are lower division students, community college students. Moose just finished his very first year of college, and I think that just helps contextualize just how amazing their work has been. Wyatt would like to know, what is the motivation to reduce the mirror size, Moose?
So the motivation behind it was originally our goal was we want to have a mirror size that does not leave any wasted area, because what that does is when you have a bigger mirror, it decreases the scanning speed. So since, let's say if you mount that device onto like the car, for example, if we have a bigger mirror area, it'll still be able to capture all of the light beam. But since there's that wasted area involved and that's not being used, it will decrease the speed of the scanning. So that's the drawback between those two parameters.
Awesome. Mimi would like to know, how did you actually gather the data in this remote format?
Oh, okay. So what me and Xiaoxing did was we had we just made a model of our setup, and so like I showed before, we just had the mirror that was rotated at an angle, and since we had that beam that was incident, we were able to use like a trigonometric relationship that relates the major axis of the ellipse to the angle of rotation. So by using like a sine function, we were able to determine how that major axis length changes with that angle.
Awesome. I think Austin has a question.
Oh, hi, thank you. Can you talk more about why your angle range is only limited to 150 to 90 degrees for your first and second layer?
Okay, I could actually, I could share that again just to show you. So the way it works was that, let me move it here. Okay, so the reason why we had you know, those specific angle boundaries was because like during our research, we found that at a theta equals 90 degrees for our mirror 2, the width, or I mean the height of the rectangle is equivalent to the major axis of the ellipse. So realistically, at this angle, the major axis of the ellipse is supposed to be the same as the height. But as we you know, increase you know, this angle, what it does is that the height remains the same, but the ellipse begins to rotate. So the major axis and the minor axis change its direction, but it's still able to be in between that original height that the ellipse was at theta equals 90 degrees. So it was, but other than that, it was just a preference of, you know, just which angles boundary that we wanted to capture.
Yes.
Do you know how the system would be powered in the automatic car? Would it just use the car battery, or would it have its own separate system of power?
I think I'm not too sure how it would be applied into like the vehicle or for whatever it's being mounted to, but I think there would be, it would be separately, or it'd be mounted separately with its own source, but I'd have to double check on that.
All right, thank you.
And B has your final question for you.
I want to ask, like, how challenging it is to produce the precise approximation for the mirror 2 that you are showing here.
Yeah, so since we had a more mathematical approach to how we solved our mirror area, we weren't able to like manually find out, you know, the size of the mirror or the shape, because since, you know, there's like an infinite amount of ellipses that could be between that angle range, some parts like on this side it would be more rounded, and then over here it'd be more vertical. So just because, you know, for the angle range that we chose, this were the orientations of each ellipse. So in that case, we would have to use like a computer program that would actually be able to more precisely map out those ellipses.
Great. Thank you very much, Moose. With that, we are going to move along to Rosario Martinez, who worked in the lab of Professor Sean Shaden this summer.
Hello, yeah, okay. Do you just see my presentation?
Yes, it looks perfect.
Okay, cool. Okay, so hello everyone. My name is Rosario Martinez. I am from Long Beach City College, and I worked in Professor Sean Shaden's lab, and my mentor was Fanwei Kong. And we worked on an automatic data processing pipeline to reconstruct coherent 3D image volumes from cardiac cine MRI data. So cardiovascular disease is the leading cause of death worldwide, and as such, there are a lot of studies to assess a patient's cardiovascular health. And computational fluid dynamics, or CFD, simulations of the left ventricle flow, along with patient medical imaging data, can lead to a better understanding of cardiovascular disease, and a better understanding will lead to patients having improved diagnoses and treatments. So the model reconstruction pipeline for CFD simulation shown here includes surface reconstruction in 3D and segmentation, which when done manually is typically a time-consuming process. However, deep learning-based methods can speed up this process. So the Shadow Lab has a deep learning algorithm that does segmentation from 3D images. So CFD simulations require geometric models of the heart that are constructed from patient-specific cardiac image scans. So CINI, cardiac magnetic resonance imaging, or CINI-CMRI, is a conventional image modality that is used for cardiac function evaluation. It produces several time series 2D image slices that are from different locations of the heart. So this is a really common image modality, so we want to be able to use these 2D images to construct models for CFD simulation. But these 2D images have to be reconstructed into 3D image volumes before the deep learning model can utilize it. So we created a data processing pipeline that can automatically take in 2D images from patients and align them to produce a 3D image. So essentially our data processing pipeline that we worked on is creating input for this model reconstruction pipeline. So this is a quick overview of our pipeline. So it takes in 2D images from CINI-MRI. And first, the images are sorted by file name. Then registration is used to reduce misalignments. Then the images are stacked. And then the spacing is applied and adjusted between images. So the data that we use to test our pipeline is from the Data Science Bowl Cardiac Challenge data. So these 2D CINI images are from over 1,000 patients of varying ages and cardiac function. And each of these slices in the CINI-MRI are taken on separate breath holds. So a series of about 30 slices were taken across the cardiac cycle for each patient. So these images are in DICOM format. So DICOM stands for Digital Imaging and Communications in Medicine. It's an international standard that is used to transmit, store, retrieve, print, process, and display medical imaging information. So DICOM images have DICOM tags, which refer to certain attributes about the patient, the study that was done, as well as the image itself. So it'll have attributes such as the name of the patient, the ID, the age. In regards to the image, it'll have stuff like pixel spacing and slice thickness. And in regards to the study, it'll have information such as the date that the study was done, among many other attributes. So on the left here is a GIF of a single DICOM image. It is showing the axial view of the heart, and it's the one slice across different time points. So registration is needed in our case because of the misalignments that can occur when the images are being taken at the different breath holds. It can be really hard for a patient to stay consistent with their breathing for 30 or so breath holds. And in addition to that, taking an image on a 2D plane from a 3D volume will cause some of the structure to be lost. And this will in turn cause visual artifacts. So registration is the process of aligning 2 or more images of the same scene that are acquired from different views or at different time points. And so in the image here, you see that there is a fixed image and a moving image. And when we stack them, they are clearly not aligned. And so registration is the process of aligning the moving image to the fixed image, so that way we can have one aligned image at the end. And the type of registration that we used was pairwise, so that means that the images are registered 2 at a time rather than as a whole. So in order to quantify the performance of the pipeline, we artificially corrupted the volumes. So we purposely caused more misalignments than applied registration and then viewed how it was aligned back. So we did this by randomly shifting the center locations of the images. And this is done with translation. So translation just refers to shifting image coordinate and shifting an image in coordinate space by adding a specified value to the X and Y coordinates. And so translation is not a part of the data processing pipeline. It is being used to quantify the performance of the pipeline. So the software that we used to create our pipeline is Simple ITK and Simple Elastics. This is free open source software, and we use the functions through Python scripts. And we're able to visualize the image outputs using MITK, which stands for Medical Imaging Interaction Toolkit. So in creating the pipeline itself, we worked on it a little bit out of order. We did first sort it by file name, which was done pretty easily with a function in Python. And then we worked on stacking it. So we're able to stack the images by using functions in Simple ITK. And this GIF shows an image, or rather a series of images that were stacked successfully. It's showing the axial view of the heart. And the GIF is going through the stack slices one at a time. And so next we worked on the spacing. So on the left is an image that has been sorted and stacked. So as you can tell, this is a very flat image. It's just not the 3D volume that we are looking for. In order to have that 3-dimensional aspect, we had to acquire the slice thickness from the DICOM images. So we acquired the DICOM tag with the slice thickness attribute, and then we were able to apply it to the image, and then we were able to get what we wanted on the right, which is more of a 3D volume. So next we worked on registration to correct misalignments. Let me use a little pointer. So the images, this is one patient at the top and then another patient at the bottom here. So here it is 2 different views, coronal, and this is a sagittal view. And so there's no registration and then registration being applied, no registration, registration. And with the no registration images, you can kind of tell that it is misaligned and that when registration is applied that it becomes a little bit more aligned, but it is difficult to see, which is why we did use translation on the images. So we wanted to have a better view of how the registration was working. So similar to before, there's one patient at the top and another at the bottom. So this is the image from the previous slide. There's just no translation applied. So you can't really tell the difference between the two. You can, but it's hard to tell. And then we applied random translation, and it is at a range from -5 to 5. And we did test it with different ranges. So we did -10 to 10 and -15 to 15. And you can see with the random translation that the image looks more misaligned than without translation. So you can see that the registration is indeed working here. And the higher that the number goes with the range, you can see that it's getting even more misaligned, where 15 at the highest, it is the most misaligned. And so you can see that the registration is working. And so here is same thing. There's a patient at the top and another one at the bottom, except this time it's the sagittal view. So it's in this view that you can tell that the registration is shifting the slices to the left. We are not sure why this happened when the registration was applied, and it is something that we would work on next had we the time. So again, like before, difficult to see the registration working without the translation, but you can see with the range of -5 to 5 that it is working. Same thing here at -15 to 15. You can see that it's very misaligned and that the registration is working to align it back. So we wanted to be able to quantify the error of the registration. So translation randomly shifts the center of the images and uses two parameters that correspond to the X and Y direction. So we took the mean of the translation parameter for each patient that we tested this on, and then took the mean of the total mean for each translation range. And we tested this on only 5 patients. We would have wanted to do more, but we were short on time. So what we can gather from this is that there is a consistently larger number in the Y direction, and that the larger the translation range means that the higher the error was. So in summary, we created a data processing pipeline that can take into the cine MRI data and output 3D image volumes of the heart. And this pipeline included sorting, registration, stacking, and spacing, and we corrected the volumes by using translation and then calculated the registration error so we could quantify it. And then future work, we would want to address the shifting of the image slices. In addition, we would like to address the patient image folders that were unable to go through the processing pipeline, and as well as applying registration and testing the pipeline on more patients so we could get a better idea of how well the registration is working. So here are references and acknowledgements. I would like to thank my mentor family for her support. She's been very, very patient and helpful for me. And I would like to thank Professor Sean Shadden for the opportunity to work in his lab. And very special thanks to Nicole and Sam for their support throughout the program. And also thank you to the Transfer to Excellence REU program, the Center for Energy Efficient Electronic Sciences, and the Hopper Dean Foundation for providing me with this experience. So thank you for listening, and are there any questions?
Well, we have some more coming, I'm sure. I had one that I had kind of pre-written here. Was there any difference in the process comparing images from patients of different ages or cardiac health? So like, for example, did the process change if a patient had a tumor?
From what I've seen, it doesn't look like there was. We tested just, are you referring to just the registration part or just the entire pipeline, or?
I mean, really whatever I guess would be the most interesting kind of tidbit from that, I guess.
Oh, okay. I was able to run the program without registration, so just the sorting, stacking, spacing for 498 patients. It went through successfully, and 2 of them did not. But I guess that problem we came across is that we're not really able to view every single image. It's really time consuming because the way we view it is literally one at a time. So we didn't have a way to really see every specific patient. So that's why we worked with a really small number of patients. But yeah, so I'm afraid we didn't have any regards to the cardiac function from one to another. We didn't really see how that went.
Okay, thank you.
Mm-hmm. I see that there's a couple of questions. The stacking. V, can you explain the stacking process? Yeah, so the stacking process, it's like heavily reliant on simple ITK functions. So it's kind of difficult to explain because I'd have to go into the details of the code itself. So simple ITK has functions that allowed us to stack the images. I don't know how much more detail I can go into it, but I'd be happy to show you my code like after this though, so you can see specifically. And let's see, Candace, Mimi's question. Oh wait, Moose, can you explain what the registration error is? Yeah, so the registration error. So let me go back. So so the translation, it shifted the images randomly. So I'll just go with -5 to 5. So you see that the total mean of the translation parameters. So for the 5 patients that I did this on, each of them had parameters, and then I took the sum of the X and then the sum of the Y. And for each patient, and then I took the total mean of all the patients together. And so, so that we ended up with the 3.84 and 4.84, and then we got the standard deviation. I know that the standard deviation, like the higher the number is, the less consistent it is. So it's the translation range, -5 to 5, is the highest, and it is hard to tell from -10 to 10 to -15 to 15 how well the registration is done. So frankly, we just needed to test on a lot more patients to really tell how the registration was working. With just 5, it's really hard to draw a really large conclusion there. Mimi's question, can this processing pipeline be extended to other organs? Yeah, it can. I mean, it has to be with the particular pipeline that I created, it has to be with DICOM images. But other than that, it should work as long as the data is sorted in a similar fashion as well. It's not specific to the heart. It was rather specific to the way that the file structure was, and also to the DICOM images, but it did not have to be heart in particular.
Awesome. Let's do one more question from Wyatt. Is there existing software that performs this 3D translation, and how does it compare to your pipeline?
So I guess there are a lot of people who have done a lot of, I guess, 2D to 3D image registration. There is a lot of literature there. They tend to focus simply on the registration, whereas we are focusing more of the, I guess, creating the 2D to 3D images. So I think that's kind of the trouble that I found, is that there's a lot of focus in literature on the registration itself rather than creating 2D to 3D volumes. And while we did use registration, we used a very simple rigid registration. So there's a lot of other registration methods, like group-wise registration, and then there's like rigid and non-rigid. And with the software that we use, which is Simple Elastics, that is specifically for registration, there's a lot of different things that you can change within registration there. So we only use one very simple method because it was, I think, we just kind of wanted to focus on just creating the 3D volumes themselves. And I think, like I said, we would want to test the registration a lot more patients, because right now it's kind of hard to tell with the simple registration method we use how effective it was.
Great. Thank you very much, Rosario. Very well done. And next we're going to transition to Samantha Childers, who has been working in Professor Bjorn Hartman's lab on design work this summer. Samantha, you should be able to share your screen. Looks good. And unmute yourself.
Sorry, the unmute button kind of vanished for a moment there. Hi, everyone. I'm Samantha Childers. I spent the summer working in Professor Hartman's lab, and I got to work on designing a computational apprentice with my mentor, JD Zempiarsko-Pierre. And so, yeah, I had a lot of fun this summer. And so this is a quick outline slide. For those of you who have like a Zoom bubble, I would recommend placing it on the right, so therefore you're not going to miss any important information throughout the presentation.
I'm going to be going over examples of previous AI assistants, our objectives and inspiration for our research, what hypothesis we had going into it, and our interviews and what we did for our interviews, how we tested, and what kind of results we got. So popular AI assistants that you might know of are Siri, Clippy, Alexa, and Cortana. These kind of AI assistants are pre-programmed to answer certain questions and perform certain functions. They are functionally different from AI apprentices that we're trying to design and develop. And so the main difference between an AI assistant and an AI apprentice is an apprentice will learn from the user and specifically basically kind of holds data that the user gives it and use it as a reference for future interactions with the user. And the user can train it to perform certain functions or tasks. So our objective for this study was, our design study, was to better understand possible AI apprentice designs and how they could assist people with creative tasks like drawing or programming or a number of
occupations. So to understand the interactions between people and those assistants as well, to ensure that our users didn't get irritated with the AI apprentice, and to explore designs for AI apprentices that can discuss ideas with the users and learn more about their field through communication and interaction. So basically we wanted to try and make an apprentice that can truly learn from the user. So we were inspired by something called rubber duck debugging. So when some programmers try and debug a logical issue in their code and they can't find it if it's a syntax, if it's already a syntax issue, it's usually easy to find. But if it's like a logical issue, sometimes you have to figure out your train of thought for the code. And so some programmers talk to an inanimate object on their desk. Typically a rubber duck is the most common. And they just talk to it to figure out their own thought process behind the code and figure out what kind of logical issue they may have had. Another way to solve their programming issues is to simply talk to another programmer and have them analyze their code with them so that they can find any issues in their code. Sorry. I got something on the chat. All right, so we wanted to try and make a middle ground between the two, where you have middle ground between a rubber duck that won't be able to respond to you and an actual human who can problem solve with you. So an apprentice is, an AI apprentice will be able to converse with their user and help them solve issues, but may not be as adept at solving issues as another programmer. So the apprentice role works well, and we believe the apprentice role would work well for humans that are trying to understand the creative work of other humans. So we hypothesized that the AI agent would be able to elicit information from, or metadata from about their user, and that humans would be able to perform basic tasks and find some things that the AI apprentice does to be helpful. So we interviewed 4 different domains, such as programming, 2D illustration, urban planning, and writing. And our main focus was to figure out which fields are people engaging creative activities while working at a computer, since we had to navigate with everyone doing remote working. And so we mainly asked these people, hey, if you had an AI apprentice working with you as you were doing your daily tasks, what kind of daily tasks would you expect this AI apprentice to do? What
kind of functions would you want some assistance with that you would be comfortable with an AI apprentice handling. And so from our interviews, we found that programmers and 2D illustrators were more likely to engage with their more creative work while at a computer. And we chose to build a design probe targeting programmers because there's a lot of existing tools that we can just modify to utilize to create this probe. And then we had a community at our lab that we could ask to test out this code. And my mentor and I already knew some programming languages, so we figured we could incorporate that into our study. So the next step is we tried to create a formative study. And in this, we decided to act out and pretend to be the AI and figure out how people would engage with us as obvious humans pretending to be the AI apprentices. And so we would created programming tasks and recorded potential users to engage with us as as an AI, and we figured out that people were we were trying to figure out how comfortable people were engaging with and sharing their information while they were coding and their creative process. And we wanted to figure out how comfortable they were with an apprentice constantly asking questions, trying to get information from their user. And we figured we should think about how to get the thought process behind the code. So for our initial testing, as I said before, for the formative study, we ran a paper prototype. And so we pretended to be the pseudo AI. We had 2 humans, one in the role of an apprentice and one in the role of the AI, and this happened over Zoom. So one person would be a programmer and the other would just be on the Zoom call taking a look at the code as the person was sharing the screen and asking questions about it and seeing how it would function. And we found that the apprentice model did seem very good at getting explanations from the user for the creative process. They were able to engage with their human apprentice and found it, typically they were very comfortable with the AI human apprentice standing, asking questions. So next we decided to try and create a design probe. And we used this by taking the P5 editor, which is an online code editor, and we designed remote human speak ability. So we meshed 2 different programs together to simulate a voice agent for this online editor. And this was so that we could have a special pseudo AI kind of interaction, which is a Wizard of Oz test. So we basically had one of our users, myself and my mentor, pretends to be an AI for another user who is using our software editor. And so for this development, we had to combine that online tool editor with the voice speaker modulator so that our users felt that they were actually interacting with an actual AI rather than a human pretending to be an AI. So how this would work is that the user would be working on the computer on the code and would share their screen with us. And we would also be able to see their code through our online editor. And we would type out responses to their questions and we would post them to their wizard interface, and it would create a robotic auditory message that would go straight to the participant so that they would think that they were interacting with an AI like Siri or Alexa or Cortana. And so this was to try and see what kind of expectations they had that they wouldn't be able to explicitly state, and some kind of, and to understand their thought process behind their code. And so this is the view of the P5 Java editor, JavaScript. And so on the left, you can see the code, and on the right, you can see a function of the code. We asked our users to create functions using a Fibonacci sequence and create like a visual using it. And so this particular user decided to make confetti circles that would increase in size by the Fibonacci sequence. Next to this slide is specifically the wizard editor. And so you can see the left, you can see on the left that you can see the same code from before and the same Fibonacci confetti. But on the bottom right, you can see the actual wizard interface where they would type out their response to questions or comments. And in this way, the wizard could directly comment on the code, like on the left, or they could just talk to the user using this interface. And I'm sorry if there's birds in the background. And so our results from this Wizard of Oz function test was that participants were willing to answer via voice to an AI agent, and participants expected the wizard to have a fundamental understanding of the program, such as their functions as for loops, while loops, what they needed to do to create spheres or rectangles. And so they would ask questions about the interface, and most participants didn't find the actual goal of the agent particularly helpful in the creative task. Like, our goal for the agent was to help them with comments or understanding just different aspects of the code. And since we ran kind of short studies, there wasn't time to create very complex kind of code where you would need comments to remind yourself about what different functions you would have to create. And so some of our participants wanted more control over their comments, where they placed it and how they were placed and what they were about. One of our veteran participants wanted to use their comments to remind themselves if they ever walked away from the computer or did other stuff. One of our participants, instead of actually creating a Fibonacci sequence, kind of made a smiley face. And so they found the comments of the wizard helpful in figuring out which part of the code was referenced to which part of the face. And so it was a very much mixed response. And so here are some of our references that we use to inspire our work. And I would like to acknowledge and thank the Transfer to Excellence Research Experience and for Undergraduates program staff for their support. Thanks to the Hopper Dean Foundation for funding this work. Thank you to my mentor, JD, and my private investigator, Professor Hartman, for their help and contributions to this research experience. And I would like to thank my fellow interns for all of their continued support and interactions. You guys have been great. So thank you guys. Are there any questions?
Well done, Samantha. Zara is wondering why specifically a Wizard of Oz probe?
Oh, the Wizard of Oz probe was to be more, I guess, to get more of the actual expectations of a user. For example, when you're specifically asked a question about what do you expect an AI to do, you might give a different answer than actually interacting with an AI and asking it questions about what you want it to do. So this was to get more of a, I'm trying to figure out the right word for it. It's like not subtext, but the unstated expectations of what you want it to do. Oh, all right, there are already questions on the chat. I'll just go through them.
Awesome. Yeah, V would like to know why you asked participants to code using Fibonacci.
Oh, this was to get people to actually do loops and functions, so that the wizard would be able to actually, since it was a kind of short-term study, we had to try and convince people to use loops and functions so that the code commenter could have something to comment about, like, hey, what does this function do? Or hey, what does this loop do? That
and then figure out, hey, do you want to comment? Now that you've explained to me what this loop does, do you want me to comment what this loop does? And so this was to kind of force people to have that interaction. Let's see.
Great. And Rosario would like to know what would be the next steps for this research.
We're working on having a more streamlined interview process for the before and after questions, and maybe to have longer sessions for this AI, so we can figure out whether or not it would be too draining to have an AI apprentice ask you questions for longer periods of time than, say, an hour of just coding some random fun code, and have something with more purpose. And oh, that's what is happening. Okay.
And then from Moose, if you had more time, what changes would you make to the apprentice model?
That's a very good question. I think if we had more time, we would make it so that the wizard interface questions didn't disappear after the session, because the way it's set up is like one user would open up the web editor, and then the wizard would also open up a web editor. And so there would be two websites that have access to the same code, and that they could edit the same code, which is really cool. The only issue I think that we really had so far was after the session was done, as soon as the wizard would log off of their website, all of the data from what questions the wizard had asked, and not really their edits to the code. The edits of the code would stay itself, but all the questions that the wizard had asked would be eradicated after each session. So I think that would be one thing that we could change to fix and have that be a permanent thing.
Awesome. And then final question from Wyatt. Did you consider a model in which the user asks questions of the AI rather than the AI asking questions of the user?
We mainly wanted to consider the AI asking questions of the user, but the wizard test was to figure out which would happen more often as well. And we found that when people were initially kind of thrust into this environment, if they didn't really know this environment that well, they would ask a lot of syntax questions initially, like, hey, so what kind of, are for loops the same in JavaScript here as they are in Java? Or, hey, what kind of function can I use to make circles pop up in my box? Or what kind of functions do I have to say to make rectangles appear in my box? And so since it's a computational kind of interactive AI, it has to respond to questions and ask questions.
Great. Well done, Samantha. And thank you, JD, for your mentorship. And thank you to everyone for your ongoing participation in this symposium. We know that Zoom fatigue is real, but we are very grateful that you're here supporting our interns. Last but definitely not least, we have Austin Culp from LA Valley College, who worked with Professor Alex Zettel this summer. Austin, you should be able to share your screen whenever you're ready.
All right, thank you.
Take it away.
All right. Hello, everyone. My name is Austin Culp. I am a student at Los Angeles Valley College, and for the summer, I was fortunate enough to be an intern at UC Berkeley's Transfer to Excellence Research Experience for Undergraduates program, where I've been able to do research alongside Scott Stonemeyer and Professor Alex Zettel on nanomaterials. I'm happy to finally present to you guys what we've been working on for these past 9 weeks, which is the, presenter mode. The sub-nanometer control of low-dimensional tantalum tellurides via encapsulation within carbon nanotubes. And before we start, in case my audio ever cuts off, please feel free to type in the chat so I could turn off my camera and fix audio. Anyway, so before we dive into our research, we first have to discuss some background information like what crystals are, and the type of crystals we're interested in to further understand tantalum tellurides. So some of you may remember from Gen Chem that crystals are just any solid state material that have all its molecular components arranged in a lattice structure. And the crystals that we're typically familiar with, like diamond or table salt, usually exist in bulk form, and mainly because they have bonding in all 3 dimensions. And the bonding between atoms dictates what the structure will look like. So there are different types of crystal structures out there, but primarily the ones that we looked at for our research were transition metal chalcogenides. Now, transition metal chalcogenides, denoted as TMCs, are crystals that are composed of transition metals, which are in the D block, and chalcogens, which reside in group 16 of the periodic table. And the reason why we're interested in these TMCs is because they exhibit they tend to show very promising properties, like for nanoelectronics, like being great for energy storage, and some of them could even be classified as semiconductors, and some even superconductors. And there are different types of transition metal chalcogenides, a lot of combinations. But there are 2 forms of transition metal chalcogenides that we want to concern ourselves with for our research, which are transition metal, transition metal dichalcogenides and trichalcogenides. So going on to transition metal dichalcogenides, known as TMDs, those take the form of MX2, where you have 1 transition metal and 2 chalcogens per unit cell. And the TMD that we looked at in our research was tantalum ditelluride. So TAT2 is a very understudied TMD. Not much is known about it except for its structure. It's known to exhibit a nanoribbon structure, or a nanosheet, if you will. And if you're looking at figure 1, here you see our molecular model of TAT2. Sorry about that. And one thing to notice here about our molecular model is that very different from what we saw with diamond and table salt is you have polar covalent bonding happening here on this A-B planar axis. But once you reach the vertical C-axis, there is no bonding between these layers, and that just leaves room for very small and weak van der Waals interactions. So a good analogy for this is you have a stack of paper, and you know, if they're not glued together, which I don't know why they would be, it's relatively easy to take a sheet of paper out from this bulk stack of paper. So moving on to transition metal trichalcogenides, TMTs, those take the form of MX3, where you have 1 transition metal and 3 chalcogens in your unit cell. And the TMT that we looked at specifically was tantalum tritelluride, which, like TATE2, is also a very understudied crystal. Not much is known about it, and the only thing known about it is its structure. And TATE3 is known to exhibit a chain-like structure. And if you look in figure 3, you could actually see our molecular model of these chains. And one thing to notice here, like we did with figure 2, is that these chains, so you're looking at a frontal view, the chains are popping out of the page, or of the screen, if you will. But if you look at the interaction between the chain, there is no bonding going on there. And again, the only thing happening there is very weak van der Waals interactions. And my analogy for that is looking at a box of toothpicks from the top. It's very easy just to pluck one out of the whole stack. So it was mentioned before that TAT2 and TATE3 are both very understudied transition metal chalcogenides, and that's mainly because a lot of problems occur when trying to synthesize these materials. And specifically, 2 main problems occur. One being that the TAT system in general is very difficult to synthesize due to the fact that when you do synthesize it, it's very unstable to exist in air, so it becomes rapidly oxidized. Your sample vaporizes the moment it's synthesized. Another problem that occurs when trying to synthesize these crystals is that both TATE2 and TATE3 have very similar synthesization routes, making it very resulting in mixed products. So going more into depth, say you want to study just the properties of TATE2, and you take the necessary procedures to synthesize TATE2, very likely are you also going to be synthesizing TATE3. And when you look at your sample and trying to analyze TATE2, it's very difficult to do that when you have a lot of background noise going on. And so these problems ultimately induce the question that is the whole purpose behind our research, which is how do we overcome these issues? So before we, for starters, we first have to approach the rapid oxidation problem. We can't look at structural properties or other problems that the TA system exhibits if our sample keeps vaporizing the moment it's synthesized. And so when looking for possible solutions, it was found actually in Professor Zettel's lab that carbon nanotubes have been able to encase other TMCs, protecting them from oxidation. So we figured in our lab that this method would work possibly for our TATE system. And not only did it worked really great, mainly because it protected our crystals from rapid oxidation, which allowed for easier sample handling and preparation for analysis, which also gave access to other characterization methods that have not been done before. And my best analogy for this is you have a very delicate painting that could be very likely to be ruined if it's out in the open. So the best way to keep it safe is sort of putting it inside these plastic tubes, where in an analogy the crystals is the painting and the plastic tube is a carbon nanotube. So why the method of encapsulation of carbon nanotubes work is mainly because, as mentioned before, when we notice that these layers do not have bonding, or these chains do not have bonding between them, it's very easy for these nanomaterials to sort of strip off from their bulk structure and sort of approach the needed lower layer or chain limit that is necessary to be encapsulated within these nanometer wide carbon nanotubes. And yeah, it's able to sometimes even reach down to a single chain or a single layer limit. So a brief overview of how we synthesize these crystals within carbon nanotubes is that we had shots of tellurium and tantalum, as well as some carbon nanotubes, all stuffed inside a vacuum sealed ampule, which is sort of like a test tube, but sealed on both ends. And then we used a chemical vapor transport, which when heated inside a furnace would have tantalum and tellurium enter this vapor phase, react with each other, and have this elementary steps, and then sort of enter the carbon nanotubes. And then when it cooled down, when gradually cooled down in a week-long process, the tantalum and tellurium begin to solidify and then crystallize within the carbon nanotubes until it reaches room temperature, and it's now this crystal structure, or these crystal structures, TATE2 or TATE3. And once we have our samples, we then apply them under electron microscope, usually specifically STEM imaging. And why we do this is to confirm whether our sample is, whether we synthesize TATE2 or TATE3. And how we do that is we look at through electron microscope and see what the structure is. And this is all done at NSEM at Berkeley National Lab, which is up the hill from UC Berkeley. So sort of a brief overview of how STEM imaging works is that there is an applied electron voltage from the electron microscope that passes through a lens. And what that lens does is it focuses the electron beam onto a single point in the sample. And because the sample is relatively heavy, tantalum and tellurium relative to an electron, the electron, when having collisions with these heavy metals, are going to scatter, and they're going to hit a detector. And so the detector sends these signals onto a computer, which basically creates this STEM image where there is greater contrast for a greater scattering angle. And the computer then saves these STEM images as an SCR file, sort of like a JPEG, but with more information packed into it, like specific features like the width and the length of the STEM image picture that was taken. And after we have our STEM images, we then use a program called Fiji, which has necessary functions like conversion factors for nanometers and 2 pixels ratio, it allows us to do things like draw a line onto our STEM images, and that line we could then plot a gray value or contrast over distance graph. And there we could see, where there's a great where along the line is there a great increase in contrast, and then a great decrease in contrast. And then taking the difference, we can then find the width of the carbon nanotubes. So here are some structures that we find to determine whether or not we're looking at TATE2 or TATE3. So what you're looking at here is actually the STEM image of the nanoribbon TATE2. This is the primary structure. And as you can see, I think you can even see the specific atoms in here. This structure definitely is very interesting and is very similar to our molecular model that we've created for TATE2. So this is the primary structure that we look at. And if we see this, we know that our sample is basically TATE2. Now, we have noticed some properties that that can be exerted. Is that when you have sometimes an extra nanoribbon will be slipped into a carbon nanotube, and there will be two nanosheets sort of inside the carbon nanotube. And when they are misaligned at a certain angle, they tend to create this moire pattern, as you see down here in this picture below. So that's just a some other slight images of TATE2 that we see, variations. Now, confirming whether the STEM image is TATE3, how we do that is if we look at the sample and we see this chain structure. And I think even in this STEM image, you could see the individual atoms. Here in the center, you see tantalum, a brighter contrast dot, because it is a heavier element. And on the sides, you see trailing attached tellurium atoms. And this again models this is very similar to our molecular model. And we have noticed some unique properties that the TE3 chains tend to exhibit when you approach this lower chain limit around 4 to 3 chains, where because there are lesser amount of chains, there's also less van der Waals forces interaction. So that actually allows the chains to start wrapping around each other in a helical manner. Okay, so here we have different nanoribbons. So we've seen different nanoribbons and chains, and we've shown that we can isolate them. Now we want to look at the parameters that can control what structures we get from synthesis. So the two parameters that we looked at for trying to synthesize these crystals are the carbon nanotube widths and the reaction temperature. So looking at this first graph, it's a box plot that shows the range at which the range of diameters each structure was found in. And comparing the box plot for nanoribbons and chains, we see that relatively, there's going to be for larger carbon nanotubes, there is going to more likely be a preference for nanoribbons, and slightly smaller diameters, there's going to be preference for straight chains. But overall, when looking at these box plots, there's a lot of overlap as well. So you can also, it is more generally more of a stronger argument that there is no correlation or very little correlation between, the diameter of the carbon nanotubes and the system's preference for structure. However, that isn't the case for when we look at the reaction temperature. When we plot the graph, this bar graph of frequency for every structure at each individual reaction temperatures, we see that for temperatures less than 600, neither nanoribbons or chains are preferred. However, there are some chains, but it's not a great amount relative to the rest of the graph. But when we reach this 600, 700 range, we see that straight chains become a preferred structure in the synthesis. And we have around 15 chains in our sample that we've looked at. So moving on to 700 or temperatures greater than 700 degrees Celsius, there's a great drop in straight chains, and the system is drastically prefers to have nanoribbons. And basically, what could be said from this graph is that definitely the reaction temperature, which the synthesis is held at, is a definite factor for the preference of the TATE system. So what these graphs and what our data shows is it actually gives some insight onto the differences between the activation energies for forming TATE2 and TATE3. Relatively, it says a lot that TATE2 or nanoribbons tend to have a greater activation energy than that of chains. It also provides a general idea of structural stability for the different crystals formed. You know, some nanoribbon structures being more stable than that of chains at higher temperatures. Now, mainly what our research has done is it's paved a roadmap for anyone who wants to desirably want to look at these at the TATE system individually and examine it in greater detail. It's given them a guideline to look at so they could further study these tensum tellurides in greater detail and seeing what kind of unique properties they hold. I would like to say thank you to my mentor, Scott Stonemeyer, and my PI, Alex Zettel. I would also like to say thank you for the TTE staff and interns. Without you guys, I feel like the support that you guys have given would not have been possible. Also making this research not been able to be possible. I would also like to say thank you to E3S and NSF for funding this research. And yeah, that concludes what we've been working on this summer. Thank you. I hope you guys enjoyed. Are there any questions?
Very well done, Austin. Moose has a question. Moose, do you want to ask it out loud?
Oh yeah, I can. I just want to ask, is CNT the only method that could be used to analyze the TATE2 and the TATE3?
Yes, from what I know so far, because trying to analyze it without carbon nanotubes, it's most likely going to be exposed to air. So I guess, it would have rapid oxidation. Although you could use the method of like trying to synthesize these crystals within a vacuum, but that still hasn't been looked at yet, from what I know.
Any other questions for Austin?
I have a question. How did you gather all the data for the graph, the temperature, frequency without the lab access?
So what happened was the plan was for me to actually go into Berkeley and do the lab procedures myself, but because that is currently unavailable at the moment due to COVID, my mentor Scott Stonemeyer actually was the one who generated these samples and took these STEM images done at NSEM. And he actually, so what I've been doing is what happened was he sent the STEM images over to me, and I'm the one who used the Fiji program that's mentioned before, and started doing these line measurements, applying grayscales, looking at the measurements of how wide these carbon nanotubes width are. And I think we've had around 600 samples, but due to a lot of blurriness in the electron microscope, we've only been able to do a few, but there definitely were a lot of samples done, and there was a lot of intense research on my part trying to take measurements for these carbon nanotubes. But yeah. Are there any more questions?
Okay. Well, that concludes our final research presentation. I want to give a huge round of applause to all of our interns. You've done amazing this summer, and I am so proud of you. In the remote format or not, what you've done is amazing, but to have done it all during a global pandemic from across the state and for Rosario across the country is very impressive. Thank you so much to our PIs and our mentors who have supported the interns and been so flexible and generous with their time to help make this possible, and for joining us today to cheer on your interns. I hope that you feel some of the pride that I feel. And thank you just to everyone for joining us and supporting our interns as they share what they've been working on this summer. I hope you all have a great afternoon and a good end to your summer.
And can we get a little round of Zoom applause for Nicole as well for running the program?
And Sam for helping so much. All right, okay. And lovely things happening in the chat. Congratulations from Ted, who's a 2019 intern. Outstanding from Dr. Perry. Thank you to the Jabeth family for joining us from Ventura. That's very exciting. Just well done, everyone. And I hope you have a good afternoon. Take care.